For that introduction. Just to clarify, I'm presenting work that I did at Cambridge as an MPhil. My dissertation was supervised by Sarah Franklin, and it focused on the sociology of parthenogenesis research. And there I found scientific language that suggested unconventional, even radical interpretations of sex and parenthood. I hope to contribute to the feminist tradition of paying close attention to word choice and metaphor in scientific descriptions of egg and sperm, nucleus and cytoplasm, gene and environment. So I'm thinking of like Emily Martin, Judith Butler, Ann Fausto Sterling, big shoes to fill. But I think that the case of mammalian parthenogenesis is particularly well suited to this kind of analysis because parthenogenesis literally translates as virgin birth. And that's, obviously a common theme in religious myths, science fiction, lesbian and feminist imaginaries, and sensational news stories, as we'll see. So virgin birth enters a laboratory setting through biologists' use of the term parthenogenesis to describe various forms of development. And that's what I'm going to be focusing on today. So let me start by breaking down this title. Parthenogenesis, as I mentioned, is a Greek word for virgin birth that scientists have used to describe eggs that begin dividing and developing without fertilization, that is, without any contribution from sperm. And this is actually a very common type of reproduction in many insect, reptile, and bird species, even in like sharks and boa constrictors. But when scientists began culturing mammalian eggs in a petri dish in the early 20th century, they also observed spontaneous division without sperm. For example, here's a picture from the 1930s that shows a rabbit egg that's began dividing and developing without sperm. And so scientists called this rabbit egg parthenogenic, or a parthenote. Later, once nuclear transfer technique was developed, scientists removed the sperm nucleus from fertilized eggs, and they also observed division after that. So this was called parthenogenic or gynogenic. So these dividing eggs would begin developing normally, but eventually something went wrong, development stalled, and they were aborted or turned into tumors. They never made it to birth. And this was actually an unexpected and strange result to a lot of scientists, given that parthenogenesis is such a common type of reproduction in many other species, but it wasn't happening in mammals. So there was a lot of research done in the 20th century asking the question, why isn't mammalian virgin birth possible? By the 1980s, scientists have described an epigenetic mechanism called genomic imprinting, and that explained why an egg nucleus and a sperm nucleus are needed for complete mammalian development. And that brings me to the focus of my talk today, this mouse. So in 2004, a group of Korean and Japanese scientists reported the birth of this mouse with 2 mothers and no father. And this was a big deal because it was the first healthy mammalian animal produced by parthenogenesis, which was formerly thought to be impossible. So just like Dolly the sheep was given her own name, this mouse was given the name Kaguya, after a Japanese folktale princess and the subject of a really excellent Studio Ghibli film that came out this year. So just like the Princess Kaguya, shown here, was born from a bamboo stump, and that's why the mouse is in a bamboo stump. But Kaguya the mouse was the product of an experiment. So I'm going to go into a little more detail about that experiment. So the scientists behind the Kaguya experiment wanted to discover more about that epigenetic process called genomic imprinting. And they were particularly interested in an imprinted gene called H19. So they engineered a mouse that had absolutely no H19, and they took an egg from that mouse and took its nucleus, its DNA, and inserted it into a typical mouse egg. So then that bimaternal egg began dividing, but the novel part was development continued completely normally. The mouse was born healthily, grew up, mated with male mice, and was even able to have her own babies. So that's a picture of Kaguya with her own babies, the typical way. So as you can imagine, the media had a total field day reporting a virgin birth, and here are just some of the headlines. And there was a lot about men aren't necessary anymore, lesbian couples can have babies. But then, of course, there was backlash, and this Washington Post headline really emphasized that this isn't safe. A lot of them mice died, a lot more than were born. Kaguya wasn't the only one, I should say. Similarly, scientists interviewed by reporters said that this experiment was not at all applicable to human reproductive technology. It was rather all about this epigenetic mechanism called genomic imprinting. And here is Tomohiro Kono, who was the lead scientist behind the experiment. And he told reporters that the idea of human parthenogenesis was, quote, senseless and, quote, an outrageous suggestion for practical and ethical reasons. And in fact, I've highlighted here, this is their abstract from the 2004 Nature paper. It reads, these results suggest paternal imprinting prevents parthenogenesis, ensuring that paternal contribution is obligatory for the descendant. So when I read this, I was really interested in a contradiction. Basically, they're reporting the birth of a parthenogenic mouse, which is this huge sensational thing, but then also saying that these results suggest that parthenogenesis is impossible and the paternal genome is still necessary. So I was like, how do I make sense of this? Is this just backlash against sensational press? Is this a real fear of fatherless reproduction? Or did all scientists interpret Kaguya's birth this way? So to find out, those are my questions, I used Thomson Reuter's Web of Science to search all the peer-reviewed scientific material that cited that 2004 experiment. So this is a citation map of the 234 publications. And as I read each one, I looked especially at how they cited the paper, in what context it was used, how they described parthenogenesis, and how they used these words maternal, paternal, and bimaternal. I am very excited to share what I found in these articles. There was really a mixed interpretation by all those scientists, but I found even when they distanced the work from human reproductive technologies and didn't mention, lesbian couples having babies, like there was no mention of that, but there still was really unconventional interpretations of sex and parenthood and a real redefinition of virginity. So I'm going to share that with you now. But let's talk a little bit about what the mixed reactions were, or what the mixed interpretations were. So sometimes authors would say, you know what, Kaguya was born, mammalian parthenogenesis is now possible, and the paternal genome is not necessary for mammalian development anymore. Or they would say, you know what, naturally, like in the wild, parthenogenesis isn't going to happen, but with extensive intervention, like in the case of the Kaguya experiment, it can happen. The third interpretation is the most interesting to me, and that's the contradiction. So they would state, viable mammalian parthenogenesis is impossible, the paternal genome is necessary. And then they would cite this experiment of the mouse with 2 moms and no baby. And here are just some examples. So you can see there's very similar language. Parthenogenic embryos die, mouse parthenogenic embryos are unable to complete full development, et cetera, et cetera. This was often in the background or introduction to a paper that used parthenogenic embryos, and so this was sort of a throwaway comment, and they would just cite the Kaguya experiment after these statements. So, how to make sense of this? One way of making sense of this contradiction relies on subtle distinctions between paternal, father, and sperm. So recall that one of Kaguya's moms lacked a functional H19 gene. And this genetic manipulation actually made the epigenetic profile of the egg very similar to the epigenetic profile of a typical sperm. So basically, the paternal imprinting was necessary for Kaguya to be born, or the paternal genome, but not necessarily a sperm. So they're describing eggs, a female genome, to have a paternal imprint, which I think is pretty rad. So let's look at sort of the results of, or consequences of this kind of language. So one, scientists are describing an egg that can mimic, you can see the language here, mimic or simulate a necessary male contribution. Basically, these scientists are describing the sex of the genome as something independent of the sex of the gamete, the egg or sperm. And so I see this as imprinting pattern being used as a new kind of sex characteristic. Sex characteristics, whether hormone levels, chromosomes, genitalia, fat and hair distribution, internal reproductive organs, we know that sometimes they don't all align, like in the case of intersex individuals or those with disorders of sex development. And so I see here, these eggs also have sex characteristics that don't quite align. The eggs have a paternal genetic contribution, and this really, I think, defies the biological basis of a simple two-sex system. Next, there was also an emphasis that this maternity or paternity of a gamete can be manipulated. So the language is, I really like this one, paternalization of the maternal chromosome. it was very explicit. The sex of gametes can be controlled, or the female genome can be remodeled. And this idea implies further scientific control over sex beyond hormone replacement or, surgeries. And it really challenges the alignment of father, paternal, male sperm, and the assumption that they are interchangeable terms. Finally, many scientists, especially those behind the Kaguya experiment, have since excluded bimaternal reproduction from parthenogenesis. So though the group originally referred to parthenogenesis in that 2004 Nature paper, the big one, that was like part of the title, later publications by the team refer to Kaguya and similar mice as bimaternal conceptuses, as opposed to parthenogenic mice. And several of the publications clarified that this was a very intentional redefinition. And you can see in the graphic there's parthenogenic and then there's bimaternal, and those are different things. They also used, although not shown here, they also used the phrase sperm-free instead of parthenogenic. So I think a literal interpretation is kind of a stretch, but parthenogenic does mean virgin birth. A literal interpretation of this distinction would be that reproduction involving two females is not virgin, an idea that suggests sperm and male penetration are not necessary components of sexual reproduction. This redefinition also just reveals the absurdity of trying to use a word like virgin, which is so steeped in cultural meaning, to describe a group of genetically modified experimental organisms. So Judith Butler, Donna Haraway, Emily Martin, all of them and many others have written about how the language of biology participates in cultural sedimentation and myth. In this presentation, I've argued that definitions of parthenogenesis are inextricably bound to definitions of virginity, maternity, and paternity. Additionally, the scientific language around parthenogenesis contributes to societal beliefs about what kind of reproduction is natural, artificial, and impossible. When scientists describe the paternal genome as necessary and mammalian parthenogenesis as impossible, the myth of Adam and Eve, a heterosexual couple with their biological children, is maintained and naturalized. When scientists describe eggs as sperm-like or mimicking a paternal contribution, this naturalizes the idea that two-mother families are in fact impossible because one woman must act like the man or the father. But on the other hand, when scientists describe maternity or paternity as something independent of sex and within scientists' control, science destabilizes the conflation of woman with maternal and of man with paternal. When scientists describe all female reproduction as not virginal, science challenges the necessity of males for sexual intercourse. This allows for the existence of genderqueer gametes and the idea of a family composed of two sexually experienced mothers and their biological children. The myth of Adam and Eve is replaced by a myth of active eggs and the female developmental biologist's pipette. Moreover, when scientists use the terms sperm-free and bimaternal instead of parthenogenesis, there's an acknowledgement that at times existing language, ambiguous and steeped in cultural meaning, is inadequate to describe nature. I think Kaguya exemplifies Donna Haraway's cyborg and Sarah Franklin's cyborg embryo, a trickster element that challenges established binaries like maternal-paternal, human, non-human, virgin, sexually experienced. Although we have made them, parthenogenic embryos make trouble for us, generating contradictions and revealing the inadequacy of our language and understanding of sex, virginity, and parenthood. Thank you, and I'd like to acknowledge Sarah and Helen, who helped supervise this work, Izum, who talked to me about the history of the field and his own experiments, and he recently published about IPS, so induced pluripotent stem cells, that from men and women that can be directed to make both sperm and eggs. So he's very much tied up in this kind of research, and it was an honor to talk to him. And the Fisk Scholarship supported me while I was at this beautiful place, Cambridge, and UCLA for inviting me here, and like the great honor of the prize. Thank you.